Hello everybody and welcome back. So, today we will start the module 4, which is about DNA damage, mutations and cancer. So, it is now very clear that the cancer cells or the growth of the tumor cells are because of the induction of genomic mutations, which means the induction of mutations in the genome, and those mutations are mostly responsible for developing tumors or cancers. Now, the question is why mutations, why the mutations will occur at all in DNA. So, here we will see how DNA damage occurs and how the damaged DNA plays roles in developing mutations in the genome. So, the cause of mutation is actually the alterations of the structures of the DNA. More specifically, most of the time it is the nucleobases. DNA nucleobases undergoes structural changes that induces the mutations in the genome, which in turn is the reason for cancer. Here this is what is the excessive, when we talk about cancer or tumor, that means the uncontrolled cell divisions. This slide I have shown before already. So, tumors or cancer means the uncontrolled cell divisions. Almost all cancers are due to mutations or damage of DNA. So, if this is a DNA and inside the cellular system, inside the cell, DNA, it interacts with many chemicals, with it is exposed to several factors such as the UV radiation. ROS is means the reactive oxygen species that are generated inside the cells, other chemicals. All those things, they lead to the lesions or called the damages in the nucleic acid structures to the nucleobases, and therefore, the structure of the nucleobases or the chemical nature of the nucleobases got changed. Now, what happens? Of course, we have very good repair system, repair mechanism is really sound. Those lesions or those damages are usually repaired back to the original DNA by our repair system, but sometimes or many times it does not happen. So, if the lesions are not repaired back to the original DNA, then it can lead to the several problematic consequences. One is, of course, it very often stops the replication process, which is called the cell death. The cell will be dead after some time because there will be no further replication, or it can induce mutation in the genome that causes either aging, or that is also the primary cause of cancer. So, one statistics is that around 10 to the power 4 to 10 to the power 6 number of lesions are formed in each cell per day. Every day around a million of such kind of damages or the lesions are formed in the DNA in every cell every day. So, you can imagine the total number of lesions that are forming and that we are experiencing every now and then, but we are still alive, which means most of these damages are actually repaired back immediately to the original DNA, and that functions properly. But many times inside tumor cells, inside cancer cells, that does not happen. So, prime factors that lead to the damages are UV radiation, reactive oxygen species, in short we call it ROS, or the chemicals. Now, the question is why the mutations will happen, if even if there is damage, why would mutations happen? So, this is the question we will answer in this whole module actually. So, I start with oxidative DNA damages or oxidative lesions. Oxidative lesions means when the DNA is undergoing chemical reactions under oxidative conditions. So, here comes mostly the ROS, reactive oxygen species. So, what happens inside our cells, lot of radicals, lot of peroxides are formed every now and then, because during the metabolic process and all these stuffs, a lot of radicals are generated. They are called the reactive oxygen species, such as hydroxyl radicals, it can be peroxides or the radicals of the peroxides, like that. And of course, you can understand when the radicals are formed, they are highly reactive. That is why it is called the reactive oxygen species. They are highly reactive, so they will interact with something, and DNA is very susceptible to reactions because it has multiple double bonds, it has functional groups. So, it undergoes reactions with these reactive oxygen species. I will draw some of the structures that are actually formed in DNA upon the oxidations. So, if you have here is the sugar, if you have uracil and under oxidative condition, they form multiple number of damages actually. So, we call them either damages or lesions. If I write a single sugar, I just S for sugar, and hydroxyl group is appearing here, and this is called 5 hydroxy. If this is deoxyribose sugar, or this is uracil, uracil is in RNA. So, this is 5 hydroxy uracil. Similarly, thymine forms this is CH3, OH, it forms 5 hydroxythymidine, DT alcohol, or it also forms, double bond is out, diol. This is known as thymine glycol, because this is CH2H CH2H unit, glycol unit, that is why this is called thymine glycol. Similarly, uracil glycol is there. This is uracil glycol. Cytosine, I am drawing for the pyrimidine bases first, then I will go to the purine. This is 5 hydroxy cytosine. So, these are some of the structures of the pyrimidine nucleobases that are formed due to reaction with the ROS species. They are actually been isolated, quite well studied, they are formed in the body. Similarly, if you look at the purine nucleobases, sugar, This is your adenine. If it is deoxyribose, then it reacts with hydroxyl radicals and produces here this produces a double bonded oxygen over here, and this is 8th position. If you do the numbering, you will find that this is the 8th position. This is known as 8 oxo, because it is the oxo group, deoxyadenosine or deoxyadenine. Similarly, one of the highly notorious oxidative damage for guanine is, I will start with guanine. So, this is deoxyribose guanine, radical, in. So, if this is cleaved homolytically, so this is a radical which will react basically, and this is electron deficient center. So, you will probably have, this is the most susceptible carbon. This upon one electron oxidation will produce this double bond. Here would be NH, this, and this is called 8 oxo guanine. I think you have already seen the structure of this when we have been doing the synthesis of the nucleobases, and this is one of the most notorious DNA lesion that is formed in the genome. So, this is upon the oxidations of the deoxyribose guanine. From here, if you do one electron reduction, then it is basically, it has a H, this will be reduced. So, you can think of kind of cleaving in this homolytically and cleaving this homolytically also. So, this bond gets cleaved homolytically. So, one H dot would be eliminated, that H dot will be here. So, it will be NH, and since this is going to be the double bond, something else has to cleave, it will be this. So, what you will have NH, H, and here you will have the NH with the sugar. This compound, this is called, this has a name. These kinds of lesions are usually called FAPU lesions. There, this is FAPU deoxyguanosine lesion. So, these are the some of the oxidative DNA damages that happen because of the exposure of the genomic DNA with the ROS, with the reactive oxygen species that are formed in good quantity during metabolic process. So, we are seeing that the bases will react, it will produce the change in the structure. So, question is how come it is related, they are related to the mutations. So, here it is. So, from guanine you get the 8 oxo guanine, this is the structure. Now, when you have the G that pairs up with cytosine, we know there are 3 hydrogen bonding and all hydrogen bonds are of the Watson-Crick nature. Now, when 8-oxoguanosine is forming, this is the structure of the 8-oxoguanine, because so what you are forming, one oxo group here, additional double bonded, and now what happens, it changes the orientation of the nucleobase. Two things can happen. One is 8-oxo G still pairs up well with the cytosine counterpart, just like this 1 2 3 through this end, as usual with cytosine. That is fair, then there is no problem. Or this actually can form an alternative base pair with adenine now, because of this structural change. Sometimes even this can participate. So, if it pairs up with adenine, then you have 2 hydrogen bonds in this fashion, and you see this is Hoogsteen base pairing, because both 6-membered ring and the 5-membered ring are involved. So, this is Hoogsteen base pairing on behalf of the 8-oxoguanine. And now, if the guanine 8-oxo G and adenine pairs up, then we have the problem. How? Let us say I will start with 3-membered, maybe T here, maybe T here, A, C, A. Let us say this is your DNA with a G here, it's double stranded DNA in the gene. Now, it undergoes oxidation and this forms T. Now, I call HOG instead of G, another T, this. You still have your A, C, A in the counter strand. Now, what happens? Of course, as I have said, most of the cases our repair system will find out that there is some abnormality over this DNA strand and it will repair it. Now, if not, let us say it does not get repaired, then what are the consequences? So, when further cell division happens, then this would be used as one of the primary strand, one of the template strand. If this is, let us say this is 5 prime to 3 prime. So, this would be used as one template and the 3 prime to 5 prime with A, C, A would be used as the other template during replication or during cell division. So, now if this is acting as the template, the synthesis of the new DNA strand will be G, T, this, and you get the original gene back, no problem there. Now, here if this is used as a template, when this is going to synthesize the counter strand, this will be A. Now, since there is 8-oxoguanine, if it pairs up with adenine, it will pick up another adenine over there and then this. So, here is all right. Now, you see when this goes further for the cell division, T 8-OG T plus this A. So, when this strand, this is a normal strand without any damaged sites over there. So, the repair system will not be able to see, it will take this as a perfect strand, perfect template, and it will synthesize the counter strand on it, T, T, T. Now, you see this part has got changed now. Your original DNA was GC sequence, here it has become TA sequence, and this will continue. Repair mechanism will not be able to find out because this is very normal, this is well known to the cells, all the nucleobases are known to the cells. Now, over time, what about this? At some or the other time, this would be detected by the repair system and would be chopped off, and it will take the correct strand opposite to it. So, and this will be repaired at some point. Even when, if the repair happens from here, let us say it is the same thing, it got clipped off, it will make a space here T, and in that case it will act this as the template, and opposite to A another T would be picked up. So, even when this is gone, chances is high that one T will be picked up in place of 8-oxoguanine. So, in both cases you are actually getting a gene which is different from the original. In other words, you are getting a mutation from G to T. So, this is called, original you had the G, in place of the G now you have the T here. So, this is usually, this is how we write G to T mutation, it is a single point mutation, single base mutation. So, your gene is now changed, and that is a problem. In the next module, we will see of course that how the proteins are synthesized using the gene as its coding unit. So, maybe you already know, that a 3-letter alphabet or 3 alphabet nucleotide sequence codes for one amino acid. So, when protein synthesis happens, these nucleobases in a pair of 3, they actually code for a single amino acid, and that is how a protein is synthesized, predetermined sequence. Now, you see, let us say DGT codes for one amino acid, which is maybe A prime, I am talking about. A prime is one kind of amino acid. Now, the gene has been transformed into TTT. Now, this sequence may not code for the same amino acid, it may code for a different amino acid. If it does, then your protein got changed and it may not function, and that is where the problem is. That is why the mutations are so notorious sometimes, not all the times of course, there are so many mutations going on. So, if the mutations happen in the important region of the gene, then they are susceptible to alter cellular functions and many times induces uncontrolled cell divisions. So, that is how the DNA lesions are related to the mutations. So, specifically we have talked now about the oxidative DNA lesions. Now, I will come back to another kind of problem that leads to mutations, and that leads to huge lesions formation, huge damage of the DNA. This is the UV radiation effect. So, impact of greenhouse gases, this is everybody knows nowadays that what the greenhouse gases, what the pollutions are doing to the earth or to our body as well. So, the greenhouse gases means the gases like carbon dioxide, nitrogen dioxide, and most notoriously and in large quantity are called the chlorofluorocarbons. They are used in huge quantity in various sectors of life. So, these are called greenhouse gases because they are not natural, they are actually effluent, or they are emitted from our daily used materials. So, what are their problems? When sunlight strikes the earth's surface, a part of the radiation is reflected back towards space as infrared radiation or as heat. So, most of the sunlight that reaches the earth's surface is actually radiated back up as a heat. So, it is a reflection, and that keeps the earth's temperature proper. it maintains the temperature of the earth. Now, what happens? These greenhouse gases, they actually shield the back radiation. They absorb the radiation, or they absorb the reflected light from the earth's surface mostly, and they trap the heat because of the fine particles, because of their huge quantity of presence in the air. So, they trap most of the radiations that was supposed to go back to the upper atmosphere, which we call stratosphere. So, as a result what happens? It increases the temperature of the earth, because the radiation is not moving out of the earth. So, it is getting accumulated, and it increases the temperature of the earth. So, which warms the earth's atmosphere, and since the radiation is not reaching back to the stratosphere, in the stratosphere it is getting cool, the temperature is dropping down. So, earth is getting hotter, as we know today as the global warming, and the stratosphere to some extent is getting colder. So, this is a fact that in the Antarctic Circle a large ozone hole has been observed from 1989 to 1986. So, this whole thing is related to the depletion or to the destruction of the ozone layer. The depletion of the ozone layer leads to an increase in the UV radiation, which now reaches the surface of our planet. I will show you how. This is it. So, ozone or ozone layer, it stays back in the stratosphere, which is around 10 to 15 kilometer altitude of the surface of the earth. Now, ozone is usually produced by the solar irradiation of molecular oxygen. So, while the production of ozone is through molecular oxygen, oxygen reacts with another radical of oxygen, one O2 molecule reacts with another molecule of radical sometimes of oxygen, that produces the ozone. That is how the cycle goes on in the stratosphere. It keeps the density of ozone intact there. And the ozone layer, what it does is it shields the UV radiation from reaching the earth's surface. So, the UV ray or UV radiation cannot reach our earth because of the ozone shield. Now, due to these chlorofluorocarbons, this is the scheme, or this is the cycle that is given, the chemical reactions that goes on. The chlorofluorocarbons, they undergoes radical productions, and those radicals actually cleaves the ozone into the molecular oxygen back. So, ultimately what do you get is the destruction of the ozone. And as I said that the temperature in the stratosphere was getting colder, that further accelerates the radical reactions. So, it helps the cleavage, or it helps the destruction of the ozone layer. And since the ozone layer is depleting, now the UV radiation is reaching the earth, and that is the real problem. So, here if you look at the table, this table given here is the range of the UV light. UV radiations is in the range of 200 to 400 nanometer, and it is usually divided into three different categories: UVA, UVB and UVC. UVA has the range around 320 to 400 nanometer, UVB is 290 to 320 nanometer, UVC is 230 to 290 nanometer. Now, what this radiation is going to do to our health? So, if you see out of all these categories, this guy UVC radiation, it falls in the range of 230 to 290 nanometer. Now, if you remember in the biomolecules, so the question is it going to affect the biomolecules? Is it going to affect our health? So, if you see the DNA or the protein, we have mentioned that DNA mostly absorbs UV radiation at around 260 nanometer. That is the absorbance maxima. So, which means it is in the range of that, maybe 230 to 270 nanometer or 280 nanometer, and proteins mostly absorb in the area of 280 nanometer. That is how you measure the protein concentration, by measuring the absorbance at 280 nanometer. But for protein, not all amino acids are active for absorbing UV radiation, only very few of them like tryptophan and so on, they absorb the UV radiation. So, for DNA it absorbs between 260 to 280 nanometer, and protein absorbs between maybe 270 to 290 nanometer, something like that. So, all of that range is basically within this frame, UVC radiation, which means if UVC light falls on our body, the DNA or certain proteins, certain amino acids can absorb that radiation, and therefore, they can do chemistry, photochemistry, because if the molecule absorbs a radiation, it goes to the excited state, and from excited state it can do different reactions. So, that is where the problem. Mainly the UVC radiation, which used to be protected because of the ozone layer, is now reaching the earth, and that actually damages the DNA nucleobases. So, greenhouse gases cause ozone layer depletion. Depleted ozone layer deseals UVC light. Increasing UVC dose on earth stimulates DNA damage, and the reason is DNA absorbs within that range of the UV radiation. So, primarily there are many kinds of DNA damages happens because of the UV. Primarily they are intra-strand dimeric lesions, those are formed within the same strand, two nucleobases adjacent to each other, they form a dimeric lesions, and the prime targets are the pyrimidine ones. The most amount of UV-induced DNA damages occurs on the primary nucleobases. Thymine, 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 cytosine, cytosine, thymine, cytosine, cytosine sites are more susceptible to undergo DNA damages or DNA lesions. So, now I will show you the chemistry that goes on and what are the products that are formed, and later we will see how these structural changes lead to mutations. So, if this is your thymine, this is the sugar, that is how it goes, and here you will have O, minus another thymine, in sugar O and the phosphate. I am not drawing the sugar. So, it is basically within the same strand, if you have a thymine and if you have another thymine next to each other, then there can be reactions under the UV light. Now, if you have H nu UV ray, basically UV, what is going to happen? So, they sometimes change the backbone also. So, today we are not going to discuss about the backbone, but mostly they interfere with the nucleobases part as well. So, in short, this you can write T, P for phosphate, T. If you remember your chemistry, the basic photochemistry, if you have one alkene plus another alkene, if you shine light on it, if you expose it to radiation, then it will undergo 2 pi electrons are there plus 2 pi cycloaddition reaction. And it will form, it is like this. Of course, when you are talking about the photochemistry, they are all homolytic cleavage of the bond, single arrow, right. So, it will form this cyclobutane. This is called the cycloaddition reaction, and there are 2 pi bonds involved. So, 2 pi plus 2 pi cycloaddition. Same thing might happen here, and same thing does happen here. This is the double bond, this is a double bond, carbon carbon double bond, carbon carbon double bond. So, what it forms is, it forms a cycloaddition in I am not writing the full, I am writing sugar here, phosphates here. And if you look at the stereochemistry, there can be two different kinds. One is, this is CH3 up the plane, one is CH3, one is H. H is up the plane also. For this is up the plane as well, the H is up the plane as well. And this is the major quantity that is formed. This is called cyclobutane pyrimidine dimer. Cyclobutane, because you are forming a cyclobutane ring, pyrimidine due to the nucleobase, pyrimidine nucleobase, cyclobutane pyrimidine dimer. It is happening between two of them. So, it is forming a dimer, and because of this stereochemistry, this is particularly called cis-syn geometry. So, these two are in cis orientation, that is why cis, and these two are also in cis orientation, which is if you have two of them, then you call it syn. Instead of calling both cis, the usual notation is use cis-syn or trans anti, something like that. So, it is a cis-syn cyclobutane pyrimidine dimer, which is the major lesion of DNA when this is exposed, when DNA is exposed to UV radiations, especially the UV-C radiation. So, this is one kind of treatment or one kind of reaction that happens. Second is, this, maybe this here, this, the same T, PT. Now, apart from this 2 plus 2 cycloaddition reaction, there can be another type of cycloaddition reaction, that is also 2 plus 2 cycloaddition. So, this is 2 pi plus 2 pi cyclo addition, same is this, but it has also a name. See, this is the double bond that we have selected, and on the other nucleobase we have done with this carbon carbon double bond, right. So, there is another double bond exists, that is this. Now, if these two double bonds, they form a cyclized product, that is known as, that has a name, it is known as Paterno-Bucchi reaction. Paterno umlaut, actually this is German name, Bucchi reaction, Paterno-Bucchi reaction. So, here your cycloaddition would be this way, goes here, goes there, and the product would be something like this. this would be your NH. Here is your sugar, double bond, this is thymine. Here is the thymine and the rest of it sugar, P, sugar, this with the formation of this 4-membered ring, and this ring is known as oxytane. So, intermediate basically, it does not stay long that way, it is called the oxytane intermediate. Now, what this will happen immediately, of course, oxytane intermediates are not at all stable. So, it will immediately be cleaved like this. So, there will be one OH coming in and there will be one double bond over here, that gives you this structure. Thymine is up the plane, OH down the plane, N is, and here double bond, there was a double bond already here, and there is this methyl, here is the N with the sugar, and there should be the double bond oxygen. This now, this position is your 1, 2, 3, 4, 5, 6, and this position is 1, 2, 3, 4, 4 position. So, this product is known as 6,4 photo product. So, now you can see how the structure got changed totally from a thymine individual. Upon the reaction with the UV radiations, you have a completely different kind of structure now. And this guy, it actually undergoes isomerization at around 320 nanometer. Here this way. Rearrangement basically. Wait, here if I write in this fashion, it will be N and straight this, and here will be a double bond. This, can you recognize such kind of structure? These are Dewar benzene, this is a derivative of Dewar benzene. So, this is called the Dewar isomer, it does form actually. So, this is called Dewar photo product. All are thymine. So, this is TPT 64 photo product, this is TPT Dewar photo product. They all have been isolated, they all have been characterized, and in different kinds of DNA, different kinds of genome have been exposed to UV radiations, and they mainly form such kind of structures. So, Dewar isomers, you can also write it like which in sugar, this is your N here like this. This is how we usually write the in a this conformation, the Dewar isomer. Now, this is for if you have the thymine and thymine. So, these are few of the photo products that you can get when exposed to UV radiation. Now, very interesting ones are if you have cytosine. If you have two cytosines CC, then what are you going to get? UV radiation, and of course, it is going to give you 2 plus 2 plus cycloaddition products. So, cyclized one, NH2, NH2 double bond N, here double bond N to this. Now, this is CPD, cyclobutane pyrimidine dimer. In short, we call it CPD lesion. Now, very interestingly, once this is formed from cytosines, what you have is amine group double bonded, and it has obviously lost its aromaticity. So, this makes this very susceptible for hydrolysis. Treatment of water deaminates the NH2 groups from here, deamination, quite rapid deamination actually. Once you form these, if you are doing your reaction in water, once you form this, they immediately be hydrolyzed, ammonia would be released or NH2 will go out, and it will give you this OH first and then it will tautomerize, that will give you this. And now you can see the real reason for mutation. So, this is formed. Now, can you recognize what is this, from where it is derived? This is basically uracil, P uracil, CPD. So, your cytosine is converted into the uracils. Now, if you think of DNA, I will show you later. If you think of DNA where you have the cytosine as a neighboring nucleobases, and after the exposure to UV radiation, if they are converted it to the uracil CPD, so eventually they will be repaired back and it will take as U, and of course, uracil does not exist in the DNA while synthesizing DNA. So, uracil would be over time be replaced by thymine. So, cytosine would be converted, originally cytosine would be converted into the thymine. It is very clear sense of mutations here. So, that is for the cyclobutane pyrimidine dimer, and if you do the other Paterno-Bucchi reaction, between this, I am writing here now, N, sorry, okay, N double bond. So, this can also stay in tautomeric form, right. So, it will be in this fashion to some extent. Here, NH and NH. This imine form. Now, this imine is double bond here. So, for thymine it was a double bond oxygen that was participating in the Paterno-Bucchi reaction. In this case, this imine form with the double bond, that will react with the other double bond of the other cytosine. So, this is also the 2 pi plus 2 pi cycloaddition, cyclo and Paterno-Bucchi reaction, and this will yield it will yield finally the NH. This will lead to the agitated intermediate double bond. So, here in that NH would be there. Yes, this is this double bond, and here would be the NH here, sorry, here would be the other NH, double bond O, nitrogen with the sugar, then this is as usual NH2 in this. Now, this intermediate is called azitidine intermediate. And obviously, this is also not very stable. So, immediately this will be clipped off . And it will give you NH2 here free, this is hydrogen, it's double bond in sugar, this is your 6,4 photo product. So, C P C 6,4 photo product, and further isomerization will lead to the dior. Amine in here straight, double bond over here, in sugar, in NH2 sugar, phosphate sugar. This is the dior valence isomer. So, this would be the photo product primarily for the cytosines. This will further deaminate here, this will deaminate again, I will show you. So, this is TPT, this is the phosphate deoxyribose, 2 plus 2 cycloaddition, cis-syn, this is the cis-syn. It can also be trans-syn, these two are trans and these two are syn. So, this is called trans-syn. This is another photo product that is formed along with this. Paterno-Bucchi reaction will produce that 6,4 photo lesions, followed by isomerization at 320 nanometer will produce the dior photo product. So, I have actually worked in this field. Now, if you take the example of cytosine 2 plus 2 cycloaddition, cis-syn, and it will be dehydrated to uracil photo lesions. Okay. Next lecture we will continue with this. Thank you.